My name is Angela Issa, and today I'm going to be talking with you about T cells and the role that they play in immune responses and autoimmunity. So T cells are essential players in adaptive immunity, which, unlike innate immunity, specifically recognizes and targets cells expressing certain antigens, and so it can kill infected or cancerous cells, for example. There are two major subsets of T cells, those that express CD4 receptors and those that express CD8 receptors. Now, CD4 positive cells are traditionally viewed as helper T cells, whereas they release important cytokines into circulation that can regulate the immune response. On the other hand, CD8 positive cells are traditionally viewed as cytotoxic killers who seek out and destroy infected or cancerous cells. Now, T cells all possess a T cell receptor, as you can see here, and this can be called a TCR, which, with the help of its co-receptor of either CD4 or CD8, can recognize foreign antigens and elicit cellular activation and immune responses. Now, for those of you who don't know, antigen simply refers to any molecule expressed by an infected or damaged cell or another cell that can activate an immune response. However, T cells can also recognize antigens expressed by the cells in our own body and can in turn trigger an autoimmune response against those cells. T cells come with a huge plethora of TCR specificities, including many that recognize self-antigens. So the question that we're here to ask today is, why don't we all suffer from autoimmune disorders? So why is our immune system not constantly attacking self? So B cells, another important immune cell known for producing antibodies, as well as T cells, are both derived from the bone marrow. However, T cell progenitors go on to the thymus to mature, whereas B cells mature directly in the bone marrow from which they are derived. And notch signaling is vital for this differentiation, as it directs T cell progenitors to the appropriate lineage and tells them to differentiate and mature in the thymus. And so we can see the mechanism of notch signaling involved in these pathways here. So once the thymus T cells go through both positive and negative selection, once in the thymus, T cells go through both positive and negative selection. In order for cells to make it out of the thymus as mature naive T cells, they must exhibit moderate and sustained affinity for self-antigen in order to stay stimulated and survive. So there's a very specific amount of affinity for an antigen that a cell needs in order to stay stimulated and survive. If cells exhibit too strong of an affinity for self-antigen, the cell is stimulated to apoptose. And this is mediated by a poorly understood signaling mechanism. And this mechanism is vital to weeding out cells that could cause an autoimmune response by attacking self. So this pathway is somewhat poorly understood, but in a paper published in 2014, researchers found that by knocking out the regulatory molecule BIM in T cells, autoreactive cells could be rescued from negative selection. And BIM is a member of the BCL2 family and is known to interact and form dimers with other BCL2 elements as part of a pro-apoptotic signaling pathway that involves a Cas-base cascade. And this pathway can be set off by strong self-antigen TCR signaling in the thymus. The researchers also showed that mutations in the BIM gene in the T cells of mice greatly impaired survival due to autoreactivity, as well as lymphoid hyperplasia, which is characterized by immense numbers of cells in the lymph nodes and periphery. Both of those disorders can be tied back to the lack of negative selection in the thymus due to BIM knockout. In conclusion, T cells play an extremely important role in the adaptive immune system, but can become autoreactive and can lead to autoimmune disorders due to faulty selection during their maturation in the thymus. The somewhat poorly understood negative selection pathway that kills self-reactive T cells via apoptosis has been shown to be critically dependent on BCL2 family member BIM. This was shown in vivo using a mouse model whose survival and well-being was greatly impaired by knocking out this gene. Therefore, we can conclude that in humans, proper functionality of BIM also likely plays a vital role in protecting us from autoimmune disorders. Thank you.